In this lecture, I will be talking about enzyme inhibition. What are enzyme inhibitors? Enzyme inhibitors are molecules that bind to the enzyme and prevent any substrate binding to it as to block its activity. Actually, in our daily life, many drugs are enzyme inhibitors. They may inhibit a certain human enzyme or enzyme of a pathogen. There are different types of enzyme inhibitors, and in A-level we will be learning two of those, competitive and non-competitive inhibitors. Competitive inhibitor is a molecule that have a similar shape to the substrate. That means these competitive inhibitors compete with the substrate to bind to the active site of the enzyme. As the substrate are less able to bind to the active site of the enzyme, the concentration of the enzyme substrate complexes formed are largely reduced, and the rate of reaction is reduced as well. However, the effects of the inhibitor can be overcome by increasing the concentration of the substrate. This is because the inhibitor is not permanently bound to the active site of the enzyme. So when the inhibitor leaves the enzyme is free to pick up another inhibitor or substrate. A great example of competitive inhibition is penicillin, a drug that is used for treat bacterial infections. It blocks the active site of an enzyme that many bacteria use to construct their cell. Then we have non-competitive inhibitors. Unlike competitive inhibitors, these inhibitors have a different shape to the substrate, and they will attach to a binding site on the enzyme but not the active site. In other words, they do not bind to the active site. The binding of these inhibitors to the enzyme alters the shape of the active site, such that it is no longer complementary to the substrate. As the substrate can no longer bind to the active site of the enzyme, there is no successful collision and no enzyme-substrate complexes are formed. Because in non-competitive inhibition, the inhibitor is not competing with the substrate, that means it is not binding to the active site directly. An increase in substrate concentration will not affect the effect of the inhibitor. That means the effect of the non-competitive inhibitors are permeable.